Hey there guys, Shamir here with another med learning video, and today we are going to talk about hemostasis. So before I start this lecture, I would ask you learners to please like, share and subscribe to med learning and also hit the bell icon so that you will be notified about our future videos. With that said, let's begin our lecture. So hemostasis is essentially a process in which the damage to a blood vessel or loss of blood in a blood vessel is stopped and healing process occurs via different overlapping processes. And this is a process in which platelets play a very important part. So for example, if someone has a prick on their finger or someone pricks their finger or damages a part of their body, then what happens around that damaged area? Then first of all, there are a few processes that happen in that area and the process of coagulation begins. So this is the process we are going to talk about today, and this is essentially a very quick process, and it starts almost immediately after injury, almost as quickly as like 15 seconds after injury, and we are going to talk about that. So if I move to my first slide, I want to talk about the objectives of the lecture, and today we are going to define our definition of hemostasis. We are going to describe the steps of hemostasis, and I'm going to also describe how the control of blood clotting takes place so that it does not go overboard, it does not become excessive. So I'm going to talk about that today. So if I move to my first slide, I want to talk about what is hemostasis. Hemostasis is essentially a process of wound healing or clot formation. So as I briefed you guys before as well in my introduction, so it is basically a healing process. It is basically a clot formation process. So whenever you like prick your finger, you see that blood starts coming out of that part of area. But almost in seconds after that, there is a gel or there is a little small clot forming around that area. So this is the process of hemostasis in action, and these are the overlapping processes that are happening during that process. So I think I have explained this slide to you guys and explained the process quite well. So this is essentially a process in which the damage to the blood vessel is stopped. So if I move to the next slide, I want to talk about the steps of hemostasis. So there are essentially four steps to hemostasis, and these are vasoconstriction, platelet plug formation, coagulation, and fibrinolysis. These are the processes that are happening during blood coagulation, and I'm going to talk about each of these in my further slides. So if I move to my first step, I'm going to talk about vasoconstriction. So immediately after a damage to the blood vessel occurs, then immediately what happens is that the blood vessels constrict, and they constrict via the releasing serotonin. So serotonin is like a enzyme, or it is also a hormone which is present in the body, and it helps in constricting the blood vessels in the body. So whenever serotonin is released, the blood vessels constrict, and they also release a hormone, or they also release a enzyme which is called thromboxanes, and these thromboxanes are essentially released by the damaged part of the vessel itself. What they do, I'm going to talk about in the next slide, but they have a very important function to play. So during vasoconstriction, serotonin is released and the blood vessel starts to constrict themselves, and eventually the platelets that are in the around the damaged vessels, they come to the blood vessel and they stick around the blood vessel. So if I move to the next slide, so the platelet plug formation is a process, and this is the second step of the hemostasis. What happens during the platelet plug formation is that previously thromboxanes were released, and they are like the signals, or they are like the transmitters for adherent platelets to clump to each other. So wherever there is injury to the blood vessel, the platelets adhere to each other, or they join with one another and they form a clump by joining with one another, and they release ADP. So ADP is also called adenosine diphosphate, and it is a very important molecule that is present within the cells, and it is also present within the blood, and it is released by platelets. And what this does is that it attracts more platelets to that site. So more platelets come to that site which is damaged, and they adhere to the platelets that are already there, and in this way they form a plug or a temporary seal. And this is like a positive feedback, because one platelet is attracting another, and this like mounts, and this eventually forms a system or a feedback system, that one thing is leading to a future thing, and in this way they form a whole process which is attracting the damaged the passing platelets to the part of the damaged blood vessel, and eventually they form a temporary seal. So this temporary seal is formed within like 66 minutes after injury, and this is called the platelet plug formation. So this is like the second step. So if I move to my third slide, I want to talk about the coagulation process, and this is very interesting, and this is represented here in a pictorial diagram that I have here on my slide. And if you see the top right of the diagram, you can see that thromboplastin is released by the damaged tissue cells which enter the blood. So first of all, immediately after injury, thromboxanes is released, serotonin is released, which constricts the blood vessels. Thromboxanes attract other platelets, and they come and eventually release ADP, which attracts more platelets to that area, and they form a temporary seal, which is also called the platelet plug. But here, what is happening is that the damaged blood vessels are releasing thromboplastin, which is essentially a blood clotting factor. And I'm going to talk about blood clotting factors in my future videos, so stay tuned for that. But blood clotting factors are essentially the factors which are acting during these three very important pathways. So these three very important pathways are the extrinsic pathway, the intrinsic pathway, and they eventually combine to form the final common pathway or the common pathway. So before all this, what is happening is that thromboplastin is being released by the damaged tissues of the blood, and these damaged tissues eventually lead to the activation of extrinsic pathway. Okay, there are a few blood clotting factors that are involved here, and we are going to talk about that in my future video, so stay tuned for that. But right now, you need to understand the general scenario, what is happening during the blood clotting process. So, at the other side, you can see that platelets are adhering to the damaged blood vessel lining, and this is obviously happening because of ADP and because of thromboxanes, and they eventually activate the intrinsic pathway. So these both pathways act to activate the prothrombin activator. So prothrombin activator is essentially an enzyme which eventually activates prothrombin, which is another blood clotting factor. And prothrombin activator also activates thrombin, which eventually activates fibrinogen. So fibrinogen is like the molecule which forms fibrin threads, and it is essentially very important for the formation of a plug or a clot around the damaged blood vessel. And when fibrinogen is activated, it forms loose fibrin threads. So first of all, fibrin threads which are little loose are formed, and then eventually a fibrin clot is formed, which is a stabilized fibrin clot. So this is the whole process that are happening. There are a lot of blood clotting factors that are involved during this process from different range of blood clotting factors, and these both intrinsic and extrinsic pathways combine to form the common pathway. So if I quickly move to the next slide, I want to talk about the last process of the hemostasis, which is also called the fibrinolysis. So after the blood clot is formed, the blood vessel heals, and eventually this clot is not needed anymore. So in this process, the fibrinolysis is basically the disintegration of fibrin, and fibrinolysis means that it is a disintegration process of the fibrin, or the breakdown of the clot basically. So the breakdown of the clot is the process of removing the clot and healing the damaged tissue. So a clot is basically formed to protect further damage to the blood vessel and to protect blood from escaping out of the blood vessels and to prevent blood loss. But after the wound has healed and after there is no need to form a clot, then the process of fibrinolysis is activated, or it happens. And this is activated by various activators that are present within the blood vessel, and they activate a substance known as plasminogen. So plasminogen is the substance which is activated by these activators, and plasminogen is the inactive form of plasmin. So it activates plasmin, and this eventually activates fibrin, which break down these products. So plasmin is responsible for breaking down fibrin and the eventual disintegration of the fibrin clot. So my last slide is going to be about the control of coagulation. So how is this whole process controlled that it does not go overboard and there is not like formation of blood clots all around the blood vessel without any damage or with like a very small damage to the blood vessel. So this process involves few different steps or few different important points which I'm going to discuss. So it is a self-perpetuating process, and it involves few guidelines that I have mentioned before, or the few important points that I have mentioned in this slide. So the first step is that the blood vessels are quite smooth and they do not let platelets to adhere to it unless there is like a damage to a blood vessel. So normal blood vessels are very smooth. And the binding of thrombin, so I previously mentioned thrombin, and the binding of thrombin to a special thrombin receptor on the cell's lining. Once bound, this inactivates the thrombin. So there is a special receptor on which thrombin binds, and it inactivates the thrombin which is already bound to it. So it basically acts as a counter to the fibrin clots, and it inactivates the clots in that manner as well. So the last point is that there are a few natural anticoagulants in the blood which inactivate the clotting factors. These are heparin and other natural anticoagulants which are already present in the blood. So I hope you guys like this video, and if you want to see future videos like these, then please subscribe to Med Learning. Also hit the bell icon so that you will be notified about our future videos. With that said, thank you for watching this video, and I will see you in the next video.